Two things stand out about the traits we have worked with so far, Mendelian traits. One thing is that they are discrete, or they come in two varieties. For example, you can have green peas, or you can have yellow peas, but there is nothing in between. The other thing that stands out about Mendelian traits is that the frequency or of phenotypes, or how often you find phenotypes in a population, is lopsided in favor of the dominant trait. We know that you have a 3:1 ratio in favor of the dominant trait if you look in the progeny of a monohybrid cross. So here, for example, 3/4 are yellow and a quarter are green, since yellow is the dominant trait. And so the frequency distribution, if you will, is lopsided. There are many traits that do not behave in this manner, that do not behave as Mendelian traits do. For example, you do not find only tall human beings or short human beings. Actually, human height varies over a continuous range of values. So human height is not discrete, it is continuous. And such traits that vary continuously over a range of values are called quantitative or metric traits. The other way in which quantitative or metric traits differ from Mendelian traits is that the intermediate values, and not the extremes, the intermediate values are most common. And so this distribution is not lopsided. Now, given the fact that quantitative or metric traits are so different from Mendelian traits, one would wonder whether Mendel's laws of inheritance can explain or still apply to quantitative traits. As it turns out, Mendelian inheritance and quantitative traits are compatible with each other. And to see how, let's introduce two new concepts. The first is the idea of incomplete dominance, of which we will see a lot more later on in the course. Let's say there are two alleles for a gene, and it's a pigmentation gene for wheat kernels, and R1 makes pigment, whereas little r1 does not make any red pigment. So if you are homozygous for two big R1 alleles, then you have red pigment and you are a red kernel. On the other hand, if you are homozygous for the little r1 allele, you don't have any alleles that make the pigment, and therefore the wheat kernel is going to be white in color. The het, however, has one allele that makes pigment, and therefore the het will have half as much pigment as the individual who is homozygous for the big R1 allele. And perhaps this heterozygous individual is light red or pink in color. The number of pigment making alleles, or the big R1 alleles that you have, determines how dark the color is, and therefore this number the number of pigment-making alleles is called the dose. The individual who is homozygous for the little r1 allele and has 0 big R1 alleles has a dose of 0. The heterozygous individual has a dose of 1, and the individual who's homozygous for the big R1 allele has a dose of 2. So we see that with incomplete dominance, instead of having discrete traits that come in 2 varieties, for example, yellow and green, we now have 3 varieties. And this is still a far cry from quantitative or metric traits that vary continuously. To see how you might get many more levels or gradations of a trait, we introduced a second new concept, and that is that of a polygenic trait, which is 2 genes on separate chromosomes that contribute to the trait, or in this case, in this example of wheat kernel color, make the red pigment. So let's say alleles big R1 and big R2 make red pigment. So we have gene 1 and 2, and the alleles little r1 and little r2 do not make red pigment. And that will give us 5 different doses. So if you have an individual who carries no pigment making alleles, so they're homozygous for the little r1 and little r2 alleles, they will have a dose of 0. If you have individuals that carry 1 pigment making allele, or they're het for either big R1 or big R2 but not both, then they have a dose of 1, and they might have a light pink color. If the individual has 2 pigment making alleles, so either they're homozygous for the big R1 allele, or they're homozygous for the big R2 allele, or they're heterozygous or a dihybrid for both big R1 and big R2, then they will have a dose of 2, and let's say they will have medium pink color. And if the individuals have 3 pigment making alleles, so they're homozygous for big R1 and het for the second gene, or they're het for the first gene and homozygous for the big R2 allele, then they have a dose of 3, and perhaps they are dark pink in color. And finally, if you are homozygous for both big R1 and big R2, giving you 4 pigment making alleles, then you have a dose of 4, and you will have the darkest red color that is possible. So with incomplete dominance and just a single gene, we had 3 levels or 3 gradations, and now with 2 genes we have 5 levels or 5 shades or 5 gradations of the trait. And it's easy to see that as you increase the number of genes that contribute to the phenotype, you will get more and more gradations, and it will appear as if the trait varies in a continuous manner. So polygenic traits that have incomplete dominance can explain the first difference between quantitative traits and Mendelian traits, namely the fact that the former vary continuously while the latter take on only discrete values. But what about the second difference? That is, Mendelian traits tend to have lopsided frequency distributions that favor the dominant trait. In other words, the dominant trait tends to be the most common in a population, whereas in quantitative traits, it's not the extremes that are the most common, but intermediate values that are the most common. So in order to see how intermediate values or intermediate doses are the most common or are the most frequent, let us compute the phenotypic proportions of a dihybrid cross of this wheat kernel color trait. And as we always do, let us carry out this cross separately for the two traits. And so I have big R1 and little r1 cross to big R1, little r1, and this will give me a quarter individuals who are big R1, big R1, and a half of the individuals will be big R1, little r1 or hets, and a quarter of the individuals will be homozygous for the little r1 trait. And the second trait is going to be very similar, and so I have the het big R2, little r2 crossed to a big R2, little r2 individual. This one is a 2, and as always in a dihybrid cross, we will have a quarter big R2, big R2 individuals, a half hets for the second gene, and a quarter homozygotes for little r2. And in order to determine the phenotypic proportions, we have to enumerate all the possible combinations of the genotypes that are possible between the two genes, and we can use branching diagrams to do that. So I'm going to take all my genotypes for the first trait and put them over here, and then I will take the genotypes of the second trait and synthesize all the possible combinations in a branching diagram. And I need a third triplet here. And my branching diagram is, you can be big R1, and then be big R2, big R2, or you could be het for the second gene, or you could be little r2, little r2. Similarly, you could be het for the first gene and then be big R2, big R2, or you could be a dihybrid. You could be a het for the first gene and homozygous for little r2, and the same way for the last three cases. And then we can write down these genotypes. So the first leaf is R1, R1, R2, R2. The second leaf is R1, R1, R2, little r2. The third leaf is big R1, big R1, and little r2, little r2, and so on and so forth. Next, we can use the product rule to compute the fraction of these genotypes in the progeny, or the frequency of these genotypes. So for example, big R1, and big R2 is 1/16. The next one is 1/8. The next one is 1/16. Big R1, little r1, and big R2 is 1/8. The dihybrid is 1/4, and big R1, little r1, and homozygous for the little r2 allele is 1/8. And similarly, these genotypes are 1/16, 1/8, and 1/16. Now we can get rid of the branching diagram since we have all the frequencies, and make some space to determine the doses of these genotypes or the phenotypes. And let's make a little table over here for the dose and the frequency of the progeny, or the fraction of progeny having that dose. And so dose 0 means there is no pigment making allele, so no big R's, and there's only one genotype which is homozygous for both the little r alleles, and the frequency there is 1/16. Then we could do dose 1, which means all the genotypes that have a single pigment making or big R allele, and there's 2 of them. There is this individual, and there is this individual. And now we can apply the sum rule, since you can only be one of these individuals at a time, and we would like to know the probability of being either the first individual or the second individual. We can sum the probabilities, and 1/8 plus 1/8 is a quarter. Then we can compute dose 2, and dose 2 means 2 pigment making alleles, and we have 3 such individuals. We have this individual who's homozygous for the big R2 allele. We have the dihybrid, and finally we have the individual who is homozygous for the big R2 allele. And if you add up these fractions, 1/16 plus 1/16 is 1/8, and 1/4 plus 1/8 is 3/8. Then we can do dose 3, and dose 3 means 3 pigment making alleles. So this individual as well as this individual, and 1/8 plus 1/8 is a quarter. And that leaves us with just one dose, which is dose 4, or 4 pigment making alleles. There's only one genotype that has all the big R's, and its frequency is 1/16. And so we see that the intermediate value, dose 2, is the most frequent genotype, and this resembles quantitative traits, this hallmark or feature of quantitative traits. And having worked these genotypes out, it's pretty clear why that is the case. That's because the intermediate values are produced by heterozygotes, like this individual, and heterozygotes tend to be the most common in dihybrid crosses, because there are 2 ways to make a het. You could get the big R1 from the father and the little r1 from the mother, but you could also get the big R1 from the mother and the little r1 from the father. So there are more ways to make hets, and hets tend to be the most common. And that is why if you have incomplete dominance and multiple a polygenic trait where multiple genes contribute to the phenotype, you tend to get frequency distributions of this sort, where the intermediate values, the intermediate doses, are the most frequent and more common, and not the extremes like they are in Mendelian traits. And finally, here is some practice. How many doses will a quantitative trait have if 5 genes contribute equally and additively to the phenotype? So we worked out this example with 2 genes, and now you need to determine how many doses, how many levels, gradations will you have if 5 genes contributed equally. And a second part of this practice problem is what fraction of the progeny of a pentahybrid cross, so like a dihybrid cross but with 5 genes, will have dose 1.